Okay, so this is the first of a series of unit videos on genetics. So first of all, before we begin, I'm going to be showing you how to do some problems. I'm going to recommend that you not work with friends on your genetics problems. It is totally fine to sit near a friend, work through your problems, and then compare your answers to see if you agree. But if you work through each problem step by step with someone, what can sometimes happen is you can think you know how to do it, and then you get to the quiz and you no longer have your friend there, and you can't do them individually. So my advice is absolutely have a friend nearby to check answers with, but don't work on this together with somebody. Also, you really need to keep up day by day because your teacher is going to be going over each night's homework the next day. So if you don't keep up with it, you can't get late credit for it. So to begin with, I just want a little bit of a history that the first known record of genetic studies was done in around 1865 by an Austrian teacher and monk named Gregor Mendel. And he was the gardener in the monastery, and he was growing peas for food, but then he started doing some scientific experiments with them and cross-pollinating them. If you use a paintbrush, you can actually move pollen from one plant to another and then end up cross-pollinating plants with different traits to see what their texture or what their phenotypes end up being, which I'll explain that in a few minutes, so what they look like and what characteristics they have. So he looked at 7 different characteristics. He looked at pod color, whether they were yellow or green. He looked at pea texture, round versus wrinkled, and he looked at plant size, tall versus short, and he looked at flower color, white versus purple. And he came up with 3 laws of inheritance that we still follow today, but he didn't really know the biology behind them. And it's kind of amazing that he came up with these just based on observations and math without knowing the biology behind it. And we now know why that biology works, but we'll get back to that in a minute. The one nice thing, the lucky thing for him, is that all of these traits were very simple in terms of how they're inherited. One of the things students really like to learn about is human genetics, but the problem with human genetics is that most of our traits are what we call polygenic, which means controlled by more than one gene, and so they're never quite as simple as a lot of the Punnett squares that you guys learned about in middle school were. So it's kind of hard to do Punnett squares with your own traits. So we spend a lot of time doing Punnett squares with pea plants and guinea pigs and the like. So anyway, Mendel's three laws. The first one is the law of segregation, and he called what we would call alleles or genes factors because they didn't know what DNA was back then. So alleles form a gene that control a trait and occupy the same location on homologous chromosome pairs. But remember, he didn't know what homologous chromosomes pairs were or even what DNA was. But he decided what he called the biological mechanisms factors. And he said factors segregate during reproduction and recombine in the offspring during fertilization. So we now know that homologous chromosomes disjoin during meiosis 1, resulting in the segregation of alleles. So this disjunction that we learned about in the previous unit during meiosis 1 specifically, is what Mendel is talking about here, but he didn't know that, of course. And that those alleles segregate 1 each to a sperm or an egg. He also came up with a law that one factor or allele is dominant and masks the expression of the other factor. And the way he discovered that is he crossed tall plants and short plants, and in the next generation, all of the plants were tall. But then he crossed those tall plants from that first generation of offspring, and then the short trait showed up again. So he realized it wasn't gone, it was just being masked by the tall factor. So a dominant allele is noted with an uppercase letter and its recessive allele with a lowercase letter of the same letter. So for example, brown eyes would be dominant to blue eyes. And it turns out actually that eye color is a polygenic trait. And all of the old textbooks that talk about big B, the brown-eyed allele, being dominant to the blue-eyed allele, it's not actually true. However, we still sometimes use Punnett squares with this trait because it's very obvious to students. Everyone knows what brown eyes and blue eyes look like. So just keep that in mind, that when we're going through this, we do know, and we want you to know, that actually there are lots of different genes that control eye color. So also he noticed that when crossing parents with two different traits, the traits are inherited independently. And he called this his law of independent assortment. And this should remind you, you should instantly be thinking, oh yeah, because of random assortment of homologous chromosomes during meiosis. So he did not know the mechanism, but he noticed that you could get different combinations of traits and that the inheritance of one trait didn't affect the inheritance of the other trait. Now, to be fair, there are traits that are inherited together, like red hair and freckles, and those are linked genes, but Mendel was lucky that none of the traits that he was really doing any experiments on were on the same chromosome, because that would have messed up all of his ratios and mathematics, and then it would have confused him. So he did get a really good basis of how basic genetics work. And of course, there are all kinds of exceptions to these that we'll talk about later in the unit. So in meiosis, non-homologous chromosomes segregate randomly from each other, and you get many combinations of genes within the gametes. So here are some terms that you need to know. You need to know what a genotype is. And when you have a zygote, and therefore a full-grown individual, you have both alleles present. You have both of each homologous chromosome. You could have two of the same dominant alleles, which we would call homozygous dominant. You could have two different alleles, which we would call heterozygous, and you could have two recessive alleles, which we call homozygous recessive. So homo means the same, and hetero means different, and zygos, or zygous, is referring to the zygote. So remember, zygotes are diploid and would have two of each kind of chromosome, which is why you have two Bs. You have either big B, little b, or little b. Now, recessive, it should be noted, is from the word recede, like to go behind something. So like recessed lighting, or receding hairline, to go behind. So basically be masked by the other one. The phenotype is the physical expression of the alleles, what you can usually visibly see. Of course, you can't actually see blood types, but brown eyes, blue eyes, coat color, if the organism is tall or short, it is the physical expression. So genotype, think of that as types of genes. And the phenotype, pH, is sort of the way the word physical, it's exactly the way the word physical starts. So I think it's the physical expression of the genotype. Now, a monohybrid cross is when we are looking at the chances of inheriting one trait at a time. With a dihybrid cross is when you're looking at the inheritance, the chances of inheriting two traits at the same time. And in this case, they would have to be not on the same chromosome, because then you have to figure out how close they are in the chromosome, and are they going to be separated by crossing over. So let's go over some problems. So we're going to do a genetic analysis of guinea pig coat color. So in guinea pigs, coat color is controlled by a single pair of alleles, and black coat color is dominant over white coat color. So if we look at the P generation, which we call the parent generation, and assume that we had two true breeding organisms, that would mean each one was homozygous. If we crossed a black-coated guinea pig and a white-coated guinea pig, and because we said they were true breeding, the black-coated guinea pig would be homozygous dominant, or big B, and the white-coated guinea pig would be little b, also known as homozygous recessive. Then we know the gametes of this individual would all have one dominant allele in them, because remember that gametes are haploid and not diploid. So you have one of each letter. The only possible allele that can be passed down by this organism is the dominant allele. This organism can only pass down the recessive allele. So what is being segregated here? Alleles. Now, the next generation, if we crossed this black-coated guinea pig and white-coated guinea pig just by putting them in the same cage and waiting until they mated, the offspring of those two guinea pig will be called the F1 generation, which stands for first filial. They would all have a black coat because of Mendel's law of dominance. Their genotype would all be big B, little b. You don't even need a Punnett square for this particular cross, because there's only one possible combination of alleles that could come together in the zygote. Now, if we crossed two individuals from this F1 generation, and we had two black-coated guinea pigs that we knew were heterozygous for black coat, their genotypes would then be big B little b and big B little b. Now it's a little bit more complex, although still fairly simple to figure out, that each of them would have a 50-50 chance of passing down either the dominant or the recessive allele. So each gamete would be either big B or little b. So now we set up our Punnett squares. Now what you need to realize about the Punnett square is that gametes, or sperm and egg, go on the outside, and possible zygotes go on the inside. So they're possible results of fertilization. So we could get big B, we could get big B, little b, we could also get big B, little b, and we could get little b. And we get that by combining the alleles row to column. Now, if we look at the three genotypes, we have big B, big B, little b, and little b. Now, when we want to figure out their ratio, we just count. We have 1, 2, and 1. Now, you always or typically put the genotypic and phenotypic ratio in order from most dominant to most recessive. That way, we all know that we get the same ratios. And all of our guinea pigs would either be black or white. The ratio here, though, instead of being 1 to 2 to 1, would be 3 to 1 because there are two ways to get a black coat. You could be homozygous dominant or heterozygous. You never know by looking at a black-coated guinea pig, unless you know what its parents or offspring look like, whether it was homozygous dominant for that coat color or heterozygous. The white guinea pig are always homozygous recessive. Here's a little bit more practice. If we cross a male guinea pig who is heterozygous for coat color with a female guinea pig who's homozygous recessive, we get a 2 to 2 ratio of big B to little b, and black to white, which is always reduced. You reduce a 2 to 2 ratio to a 1 to 1 ratio. If on a quiz you wrote 2 to 2, and you wrote this Punnett square, you'd get most of the points. You'd get the full points for your Punnett square, but you'd probably lose half of the points for not reducing your ratio. Always reduce the ratio if you can. I once had a student try and reduce a ratio that wasn't reducible, and then, you got to remember the rules that you learned about ratios in math. Okay, now and by the way, you don't even need to do this row, because if you don't do this row, it saves you a little bit of time, and you get the 1 to 1 ratio, and you could reduce before you even start your Punnett square. But it doesn't really save you that much time to skip that row if you are doing a monohybrid Punnett square. It makes much more sense to do that with the dihybrids. I'll be showing you that soon. So in this particular case, if we crossed a homozygous dominant with a homozygous recessive, I feel like that's a Punnett square almost not worth doing. That one you would probably just do in your head. And now that ratio would really be technically 1 to 0, but I've never seen that. I just usually just say they would all be big B, little b, or all be heterozygous for black coat color, and they would all be black. Now let's talk about dihybrid crosses. Dihybrid crosses are ways to figure out the probability of inheriting two traits at the same time. And they are always examples of Mendels' law of independent assortment. So alleles on different chromosomes assort independently of each other. Genes that are on the same chromosomes, though, are called linked genes, and we've talked about having red hair and freckles in humans a lot. Those are on the same chromosome. If you look at this picture of what we used to call, when I was a kid, Indian corn, but it's decorative, and people hang it on their door sometimes to say, hey, it's fall, and I'm decorating for fall. But if you look, there are two different colors of the corn seeds and two different textures. One thing students don't realize is that each individual corn kernel is actually a seed for an entire new plant. So it's kind of like a baby individual corn plant. It hasn't obviously been planted yet and hasn't grown yet, but you can see that there are a lot of purple and smooth, and that there are fewer that are wrinkled or yellow. So it turns out that the color that is dominant is purple. Purple is dominant to yellow, and smooth is dominant to wrinkled. So let's do a Punnett square. Now, if we were to cross two plants that are heterozygous for both traits, both plants would be purple and smooth. And that's because purple is dominant to yellow and smooth is dominant to wrinkled. And then the genotypes would be big C, big T, little t, or heterozygous for both traits. Students are always like, well, where'd you get that? Well, we just chose particular letters. The letters are something that I will usually give you and tell you which letter to use. If I don't, you choose, but make sure you choose letters that you can tell the difference between the capital and the lowercase. But the C in this case stands for color and the T for texture. Okay, now the reason that we went big C, big T, little t is because it said let's cross two plants that are heterozygous for both traits. So that's how we knew it was big C, big T, little t. Now the trick here, this is where it gets a little tricky, although not that tricky if you've been paying attention in your previous math classes, is to figure out what are the possible combinations of alleles in the gamete. Because the two C's are on homologous pairs of chromosomes, and so are the two T's, you get one C and one T. You go from having 4 alleles to 2 in each of the gametes, because remember, gametes are haploid. Now you get one C and one T. I've seen students do some really wonky things with their Punnett squares and have like C's all the way across the top and T's all the way down. Each gamete gets one C and one T. Now, to figure out all the possible combinations, you FOIL. You do first, outside, inside, last. So first would be big C, big T. Outside would be big C, little t. Inside would be little c, big T, and last would be little c, little t. Now, it makes it much easier when you're figuring out the possible alleles in the gametes of the second one if it's the exact same as the first parent. So if we FOIL that again, it's the same combination. Now, this means we're going to have a Punnett square with 16 squares. This is more tedious than it is really challenging. Just make sure you leave yourself enough room when you spread it out. So if we set this up, we set up all the possible gametes across the top and down the side. Then all it is bringing them together in the Punnett square. So big C, big T, big T. And we fill those in. Now each zygote inside the Punnett square is going to have 2 of each letter. Now, it doesn't matter whether I had started out with C's first or T's first, but once you pick it, you need to be consistent. One of the things that students always find sort of weird is that you see, like here, somebody will go, well, why'd you put this little c before the big T? Don't you always put the dominant one first? Well, yeah, but remember that the T is for texture. That's a different gene than the C that's for color. So you wouldn't put the T before the C because it's not dominant to that allele. It's only dominant to a recessive texture gene, which would be the T. So that's why, whatever you start off putting first, you continue to put first. So now it's just a matter of figuring out the phenotypes and counting. So this one would be purple and smooth. This one would be purple and smooth, and this one would be purple and smooth, and this one would be purple and smooth. And you have 4 different possible phenotype combinations. You have purple and smooth, and purple and wrinkled, yellow and smooth, and yellow and wrinkled. Now, there are a couple ways that you can keep track of these so that you don't miscount. Some people like to use colored pencils and use 4 different colors. The one thing I will tell you is if you do that, don't color the whole square in. You will waste your time coloring squares, and I've seen people trying to perfectly color in their squares. If you're going to do that, just color around the edge of the square. It's much faster. If you don't have colored pencils, you can pick any symbol, like a circle, a square, a triangle, and a star, and then find members that fit that trait and put that symbol in there and then count them. If you do this, you will find, by the way, that there's patterns. These will all be purple and smooth, and so will these, and then these too. So if you counted this, you would find that there are 9 purple and smooth, 3 purple and wrinkled, 3 yellow and smooth, and 1 yellow and wrinkled. That is a 9 to 3 to 3 to 1 ratio. This ratio is the ratio you will always get when you have two parents that are heterozygous for both traits, as long as the traits one trait shows complete dominance over the other. So purple is always dominant to yellow. There are some patterns of inheritance where we call them incomplete dominance. This 9 to 3 to 3 to 1 ratio doesn't work with those, so just beware. But if you were taking a quiz, you need to show your work. If you're doing a unit test, you don't need to show your work because remember, our unit tests are Scantron. So you could save a lot of time if you know this 9 to 3 to 3 to 1 ratio on the unit test. But remember, on a quiz, you would have to show that 16 square Punnett square. Now, you notice that you have the greatest number that are dominant for both traits, the least number that are recessive for both traits, and the 3 out of 16, purple, wrinkled, or yellow and smooth. And by the way, if you wanted to put this in fractional form, it'd be 9/16 purple and smooth, 3/16 purple and wrinkled, 3/16 yellow and smooth, and 1/16 yellow and wrinkled. Remember, you can't reduce a 9 to 3 to 3 to 1 ratio because it's already down to 1. You can't reduce below 1. Now, I want to talk a little bit about test crosses. If you have an individual that has the dominant phenotype and you want to know whether they're homozygous dominant or heterozygous, you cross them with an individual who's homozygous recessive. You can't really do that with people, but you can do it with fruit flies or guinea pigs typically. But if any of the offspring had the recessive phenotype, then the parent with the dominant phenotype had to have been heterozygous for that trait. So let's do a dihybrid test cross. So we're going to cross a corn plant that's heterozygous dominant for both traits with a plant that is recessive for both traits. So that means a purple smooth corn plant with a yellow wrinkled. Now the genotypes of the purple smooth parent would be big C, big T, little t, or heterozygous for both traits. The yellow and wrinkled would be little c, little t, little t. Now when you do the possible combinations of gametes, you just use our old FOIL method, first, outside, inside, last, and you get big C, big C, little t, big T, and little c, little t. Now, for these, they're all little c, little t. You don't need to make a 16 square Punnett square for this because each row would be exactly the same as the row above it. So you can reduce from the beginning and note all of those are the same, so you only need one row. Now, students always want to know, well, what if I forget to do that and I make a 16 square Punnett square? If you do that, you're going to get the same answer, although you'll get a ratio that needs to be reduced. If you do that and you properly reduce your ratio at the end, you will get full credit. You will never get those 5 minutes of your life back, but you will get full credit. So it's nice to be able to simplify before you begin because it saves you time. And why make a 3-minute problem a 10-minute problem? But in this case, when you fill out your Punnett square, you would get purple and smooth, purple and wrinkled, yellow and smooth, and yellow and wrinkled. And the ratio would be 1 to 1 to 1 to 1. Now, if you did a 16 square Punnett square, you'd get a 4 to 4 to 4 to 4. And if you got to the end of that and realized, I can reduce that to 1 to 1 to 1 to 1, and then you wrote 1 to 1 to 1 to 1 on your quiz, full credit. But you might run out of room later on your quiz because you spent so much time making a big Punnett square when you could have used a smaller Punnett square. So now I want to show you, most people do not have a hard time with what to do in this particular case. If all 4 of them are the same, most people go, oh yeah, I recognize that. And most people will give me a 4 square Punnett square instead of 16. But I want to show you an alternative possibility. So we're going to move the camera for a minute. So what if you had big C, big T, little t, by big C, big T, little t. Now in this case, you would still FOIL and you'd have 4 different possible combinations. But in this case, these two are the same. I always, if I notice that two alleles in a dihybrid are the same, circle them. So that when you're distributing, you realize there's only two possible combinations. You'd have big C, big T, or big C, little t. I'm not going to do this whole Punnett square, but I will show you how to set it up. So in this case, I know that I'm going to need 8 squares. I have big C, big T, big C, little t, little c, big T, and little c, little t on here. And now I have big C, big T, and big C, little t, because there's only two possible combinations. So if you circle any that are alike and distribute, it might save you from making a bigger Punnett square than you need and wasting some of your time. Again, if you do a big 16 square Punnett square, it's not the end of the world so long as you know how to reduce ratios. But it definitely can save you time to reduce before you start setting up the problem. And that's the end of my tutorial on monohybrids and dihybrid crosses.